Okay, we should be recording now. So, welcome to the AP Biology Unit 3 review for the second time this night. Hopefully, nothing breaks, crashes, anything like that. Uh, yeah, so let's get started. So, a little bit about why you're here tonight. This review session was prepared by Golden, that's me, as part of Five Hive. Just a little bit about us. We're a volunteer student run initiative with over 70 volunteers and over 10,000 combined hours. And what we want to do is we're providing resources written by students for students that are taking AP courses. And you can check out our website, which is fivehive.org, for additional resources and practice exams. And if you'd like to contribute to Five Hive by creating resources, hosting lectures like me, or promoting this project, you can check out this document, which is linked in the slideshow for more information. And down here are our socials, our website, TikTok, Instagram, and the Discord server. And you guys should have access to the lecture slides, but if not, let me know and I can put the link in the Discord chat. All right, so I also do ask that you guys fill out this lecture feedback survey. You can scan this QR code on screen, or you can type this bit.ly link into your web browser and just fill it out as we go along. Your feedback will not only help us improve our lectures, but we'll go through and read each person's individual feedback, or at least I will, and this will help me figure out what to emphasize on future lectures and also what does work, what doesn't work, things like that, and so on. So you'll help not only yourself but also other students who are currently taking the course and who will take it in the future. So let's go through some expectations. So this presentation, the lecture, I'm not here to help you guys learn Unit 3. You guys should have done that already, but this is intended as more like a review session. And what that means is I'm here to help you refresh or reinforce your understanding of the course, AP Biology. But also do bear in mind that I cannot guarantee that you get any specific score on your exam, which is May 5th at 8 a.m. your time, or any particular grade in your course. And just a few legal things. AP is a trademark registered by the College Board, which is not affiliated with and does not endorse this resource or the 5 team. And while you guys are going through this lecture, I ask that you engage with the lecture, and that means asking questions, participate in practice, and also discussing with each other in the Discord chat. So, what tonight's lecture is going to look like is as follows. First, we're going to get a good sense of how prepared are we for this lecture. And then we're going to do a quick topic overview of what to expect. And then I'm going to lecture a little bit about each individual topic in Unit 3. And then after each uh, sort of, I guess, group of topics, or every now and then, we'll have a couple practice questions, multiple choice in the style of the AP exam. And at the end, I'll show you where you can find some other review material if you want further practice, and then I'll take any questions that you have at the end. And I will pause throughout this lecture to take questions. So, let's get started. How prepared are you in your knowledge of Unit 3 cellular energetics, with 1, or I guess 0 in this case, being I have no idea what energetics are, and 10 is give me the exam right now? You would just put your answers in the, what is this, the lecture channel chat. Let me get a quick sense of where we are. So I'm getting around 4 or 5 with a couple 7s, so that's pretty good stuff. Um, yeah, so hopefully at the end of this lecture, most of you will be at about an 8 or a 9, hopefully some 10s. Let's see how well I can refresh you guys on Unit 3. Let's get started. So a quick topic overview, Unit 3, cellular energetics. There are 7 main topics in this unit, making it a bit less than average in length compared to all the other units, but it is also weighed a good amount. It's 12 to 16% of the exam, which is a lot more than all the other units similar in length. Like I believe some of the other units like that are only 7 topics. Some of those are only weighed like around 8 to 10%, something like that. So you can see there's really big emphasis on cellular energetics, and it's also the unit that most students would say is one of the more difficult ones, just because of how much memorization goes into it. And so we start out with enzymes. That goes into cellular energy, photosynthesis, cellular respiration, and then finally it talks about fitness, which really I think should be in, I think it's unit 7 that talks about evolution, natural selection, but according to College Board, it's cellular energetics. I don't know why. So let's get started here. So first we got to review enzymes. Now, enzyme structure. Enzymes are proteins, and basically what they do is they speed up biological reactions by lowering the activation energy. And a biological reaction here can mean all sorts of things, such as breaking down food in the stomach, or let's say creating new biological molecules. Enzymes can do basically any sort of reaction that happens in your body. There's going to be a specialized enzyme for that. And as you can see on this diagram on the right, each enzyme has an active site where the reaction takes place. And how does an enzyme determine what sort of reaction it participates in? Well, we can see that the shape and charge of the substrate, which is what the enzyme, which is what the enzyme either breaks apart or forms in the reaction, it must be compatible with that of the active site. A bit more about catalyzing the reaction. As I've just gone over, enzymes act as a catalyst for a reaction, and this means that they can speed up the rate at which the reaction proceeds by lowering the activation energy required. As you can see on the left side here, the red line symbolizes the reaction process without the enzyme, and the blue line symbolizes the reaction process with the enzyme. And clearly you can see that the energy hump for the catalyzer reaction is a lot lower than that for the reaction without the enzyme. So basically what this graph is telling us is that when you add an enzyme into the reaction, the activation energy is lowered a lot. Therefore, this reaction is faster than it would happen without the enzyme. And now in chemistry, this usually means that more reactants collide with sufficient energy to react. But we're not taking AP chemistry, this is AP biology. So in a biological context, what an enzyme does is it brings together reactants in the right orientation and provides a suitable environment, whether that's pH or just brings together certain substances in order to speed up, you could say, speed up the reaction. And sometimes enzymes even participate in the reaction directly. So now let's talk about the environmental impact on the function of an enzyme. So as we've gone over, enzymes are proteins. But what exactly does this mean for an enzyme? Let's put our answers in the chat. So what I'm looking for is if an enzyme is a protein, then what characteristics would that make an enzyme have? All right, there's a couple of good answers. All of them are correct so far. So you are all correct. The structure, the function of an enzyme is based upon its structure. Uh, and yes, because an enzyme is a protein, enzymes are in fact made up of amino acids. And this also means that enzymes can denature. So let's look at that. In Unit 1, you should have learned that a protein is a complex polymer that consists of amino acids, and the sequence that which these amino acids are connected to each other in determines how it's going to fold in space to form the final protein. The shape of a protein is what determines its function. Now, being protein, enzymes can denature, as you guys have remembered. And what that means is the enzyme, it denatures, it loses its shape. Um, you can see down here in this image that the normal enzyme on the left, its active site has the correct shape to bind with the substrate there. And so this reaction will proceed uh, after being catalyzed by the enzyme. On the right here, something's happened to the enzyme, whether that's um, whether it was too hot or too cold, it was too acidic, something like that. But you can see that the active site is no longer the correct shape to bind with the substrate. And so this reaction, this enzyme can no longer catalyze whatever reaction that is going on here. So the reaction can still proceed, but it's not going to have the added boost provided by the enzyme. And sometimes it is possible for a denatured enzyme to reform back into the correct shape. And as I previously said, enzymes are affected by temperature and pH. And pH is just a measure of, really just a measure of how many hydrogen protons there are. The exact mathematical formula, you don't need to know this for the AP bio exam. This is more of a chem concept. But pH is negative log of the concentration of the hydrogen atoms. And the relative concentrations of substrates and products determines how efficient the reaction proceeds. So if I've got a whole lot of substrate and a tiny amount of product, we're going to have a more efficient reaction here just because it is more likely for an enzyme to be floating through this environment and encounter a substrate that it can then split or combine into a product. And so as more and more substrate gets turned into product, the reaction is going to slow down. Now, higher temperatures increase the speed of molecules, such as the enzyme. An enzyme is basically just a gigantic molecule that increases the rate of reaction. However, remember that enzymes are proteins, which they can denature. So past a certain temperature, the enzyme will denature, and then the reaction is no longer catalyzed by the enzyme. And finally, about enzymes, are these things called inhibitors. There are two main types of inhibitors that you need to know for the AP exam, and those are competitive and non-competitive inhibitors. On the left here, on enzyme A, we've got a competitive inhibitor. And this means that the inhibitor is going to bind either reversibly or irreversibly directly to the active site, and it's physically going to block any substrate from binding to the active site to be processed by the enzyme. And on the other hand, non-competitive inhibitors, on the right here, we've got a non-competitive inhibitor bound to enzyme B. And non-competitive inhibitors, they bind to allosteric sites and they're going to change the shape of the active site. So notice how enzyme B's active site can no longer bind with substrate, meaning that enzyme B also cannot catalyze the reaction, whatever reaction might be happening over on the right. All right, so now that we've reviewed everything there is to know about enzymes for the scope of this course and the exam, we're going to do a quick progress check. It says, which of the following best explains how the structure of an enzyme relates to its ability to catalyze a specific reaction? If you look down at the, I guess at the purple magenta bar at the bottom, this question covers topics 3.1 and 3.2. So if you guys want to answer this question in the chat, I will also check the chat now for any questions. What use do inhibitors have? There's really biologically, I would say like naturally there isn't too much use, but in the medical field, um, if say there's a reaction, because we all know that to maintain life, homeostasis is a pretty important thing to maintain life. So maybe there's enzyme in the human body that's catalyzing a reaction is causing it to proceed in the forward direction too quickly. And so we're building up a whole lot of product that our bodies can't get rid of. So at this point, we can introduce inhibitors to stop the enzymes from catalyzing whatever reaction this is. And so we can slow down the rate of that reaction and give our bodies time to get rid of all that waste product. An allosteric site is any site that an allosteric site is basically somewhere in the enzyme where where the non-competitive inhibitor can come in and bind to the site in the enzyme. Basically, it's somewhere that's not the active site, and but when the inhibitor binds to the allosteric site, it changes the shape of the active site. There's not like a definitive section of the enzyme that defines the allosteric site. It can really be anywhere along the enzyme, just not the active site. Okay, so anyways, back to the progress check question. If you guys would care to think about this, maybe put your answer in the chat. I'll give you guys about 2 minutes, and then we'll move on and look at what the correct answer is. So it looks like all of you are in agreement that the answer is B. So let's find out. You guys are correct. So the answer to this question was the active site of an enzyme has a specific shape that binds to complementary substrates, which stabilizes the transition state and lowers activation energy. Pretty self-explanatory. Um, A is not correct because, well, the enzyme doesn't provide energy to the reactants. It just lowers the activation energy. So A is not correct. Um, C, enzymes break down during the reaction to release products. That first half of the answer is not correct. Enzymes don't release products. Some of them may break down during the reaction, but they definitely don't turn into the products. D, the overall shape of an enzyme is rigid and does not change. That first half is also incorrect, as we've seen here. During inhibition, the shape of an enzyme will change if it's a non-competitive inhibitor, and also recall that enzymes can denature. So the process of elimination or just knowing the correct answer right off the bat, the answer to this question is B. Now we're going to do another one right quick just because we covered 3 topics in a row. The scientist placed a sample of the enzyme amylase, which normally functions best at 37 degrees Celsius neutral pH, that's about 7.0, into a strongly acidic solution heated to 70 degrees Celsius. Which of the following best explains the likely effect on the enzyme's function? And this question is in relation to topics 3.1 and 3.3. All right. Looks like we are all in agreement that the answer is C. That is correct. It's also pretty self-explanatory. Not too much that can trip you. Not too much they can throw to trip you guys up in regards to enzymes. Uh, A is A could be correct at first glance, but if you look closely at the wording of the question, uh, it says that the scientist placed the sample into both a strongly acidic solution and he heats it and the scientist heats it to 70 degrees Celsius. So the fact that there's a pretty big temperature gap between 70 and 37, and that the solution is strongly acidic, should tell you that this probably has something to do with an enzyme denaturing. And then B, the enzyme's active site will adapt to the new conditions. Active sites don't do that. Whatever shape an enzyme folds into, that's it. The shape of an active site is pretty rigid. So each enzyme specializes in participating in one and only one reaction. So active sites do not adapt to new conditions. C was the correct answer. And then D, the enzyme will continue to function normally as enzymes are not affected by changes in temperature or pH. The second half of this answer is incorrect. Enzymes are affected by changes in temperature or pH. All right. Topic 3.4 is cellular energy. Is not a whole lot that you need to know for this section, but the four main key points are here. The first one is that all living systems need a constant input of energy. And this makes sense because we and all other living things need energy to maintain homeostasis, perform biological processes, things like that. Uh, and this carries into our second point here is that the energy input must exceed whatever energy expenditures we have to maintain order in our body. Uh, and this relates to the second law of thermodynamics. And obviously, you don't need to know word for word what the second law is to pass the exam. And then the third key point here is that cellular processes that produce energy, which is what we're about to talk about, photosynthesis and cellular respiration, they can be coupled with processes that consume energy, such as generating heat for warm-blooded animals, or contracting muscle cells to provide movement. And finally, loss of order or energy input results in death. Basically, if you don't have any more energy coming in, all the energy is going out, you will die. That's that's just how it is. All right. So that was 3.4, pretty short. So now 3.5, we're moving into the meat of this unit, which is also where most students mess up because there's just an absurd amount of information to know for this unit. So we're going to start out with 3.5 is photosynthesis. So here I've got a little diagram of exactly what happens during photosynthesis. And obviously, you should know by now that photosynthesis only occurs in plant cells because only plant cells have the organelles required to do photosynthesis. So, first, photosynthesis takes in water, sunlight, and carbon dioxide as reactants, and it produces ATP, which is energy for cells. It produces oxygen, and it also produces sugar, which is a plant's main food source. So first, in the thylakoids, light catalyzes the light-dependent reactions. It produces ATP and an electron carrier, NADPH, which then goes into the Calvin cycle that takes carbon dioxide in and produces sugar. And this happens in the stroma. And the thylakoid and stroma, these are just two of the organelles I can find in a plant cell. So let's take a closer look here. The light-dependent cycle, here's what happens. So this is kind of like a chronological order diagram where we're going to start on the left here. And what happens is sunlight, uh, energy from the sunlight comes in, is channeled into this green blob here. It's labeled PS2. That stands for photosystem 2. And essentially, a bunch of chlorophyll atoms bounce around this energy from the sunlight and channel it down into a central pair of chlorophyll atoms here at the center of the photosystem labeled P680. What this means is that they are most sensitive to light, that it has a wavelength of 680 nanometers, and that's a red color. And what they do is they get excited by the light, release an electron that travels like this through the cell membrane, the membrane of the organelle. It creates a hydrogen proton gradient. So it helps the energy from this electron traveling through the electron transport chain. Uh, the organelle harnesses that energy to pump a bunch of protons to one side of the membrane. And then you'll see later over here that all these hydrogen protons, They then rush back out to create homeostasis through a channel protein called ATP synthase. More on that in a little bit, but you will see that the electron released from photosystem 2 travels all the way down over here to photosystem 1. And then the energy from that electron, plus some from sunlight, it excites another pair of chlorophyll atoms here labeled P700 because they're most sensitive to light, that it has a wavelength of 700 nanometers, and that's still red. The 20 nanometer wavelength is going to make too big of a difference. But same thing goes here. Same thing happens here as it did in photosystem 2. They're going to release an electron, and we're going to harness the energy of this electron to turn NADP plus back into its electron carrier form NADPH. And then finally, the proton gradient that we built up earlier with a bunch of protons down here inside the thylakoids, they're going to rush all back out into the stroma through this channel protein called ATP synthase. And basically, what it does is imagine the protons as water, and inside this ATP synthase, it's like a little turbine wheel. So all the proteins rush out, spin the turbine, and we harness that energy to turn ADP. We tack a phosphate group onto the end, and we turn it back into ATP. And over here on the left, you'll see where do all these electrons come from? We have to replenish them somehow. And the answer to that is, well, we can take a water molecule, we split it into oxygen and hydrogen, and releases some electrons to replenish the electrons that we use to make ATP. So whatever we use, we have to replenish it, and that's how we replenish the electrons that we used. So after that, we go into the Calvin cycle. So we're going to start out with these molecules called RUBP. And we're going to take some carbon dioxide. We're going to combine the carbon dioxide with the RUBP in a 1-to-1 ratio. We're going to make this molecule called Rubisco. This step is called carbon fixation. And then we're going to take one molecule of Rubisco, we're going to split it in half basically, and we're going to form a molecule called 3-PGA. So we start out with 3 Rubisco, we now have 6 3-PGA. And then during this step, we reduce our electron carrier NADPH back into NADP plus. We take an electron from it. We take the energy from that electron, and that powers our Calvin cycle. We also take some energy from ATP, turning it back into ADP. And after the reduction step, we end up with 5 molecules of G3P. And of the sorry, we end up with 6 molecules of G3P. And of those 6, only one of them goes on to make glucose for the plant to store as energy or use. 5 of those G3P molecules are recycled in a step called regeneration. That also takes some energy from ATP, turning it back into ADP. But after regeneration, our 5 molecules of G3P are recycled back into our starting 3 molecules of RUBP. And this cycle just keeps going and going, taking RUBP, combining it with carbon dioxide, taking energy from NADPH and ATP, and turning it into G3P, which goes on to make glucose, the plant's main food source. Will we have to know every step of the Calvin cycle, like all of the subproducts and substeps as well? The answer is no. You will only need to know that realistically you'll need to know that the Calvin cycle takes RUBP and CO2, turns it into G3P to make glucose. Uh, and the energy comes from NADPH and ATP. You are not going to need to know each individual molecule name or anything like that. They're not going to ask you what is the molecule that turns G3P back into RUBP. That's beyond the scope of AP biology. So we don't need to know G3P and PGA, etc. Uh, at least when I took the test, they definitely didn't go that much into detail. The three, the main things to know is just that RUBP plus CO2 equals G3P. and 5 of those are recycled, 1 goes to make glucose. And the energy comes from NADPH and ATP. So ADP is adenosine diphosphate. It's what happens when we take one of the phosphate groups from adenosine triphosphate, which is known as ATP. And each of these phosphate groups contain energy in their chemical bonds that we can use to power cellular processes, such as this Calvin cycle here. And so ADP is what happens when we take one of those phosphate groups off of it, ATP, to use to power a cellular process. And then the ADP goes back here into the light dependent cycle, where we harness the power of the hydrogen proton gradient to put another phosphate group back onto the ADP and turn it back into ATP. So we continuously regenerate ATP that then goes to power the Calvin cycle and makes glucose for the plant. Yes, the Calvin cycle is light independent, whereas the light dependent cycle, since it starts by harnessing sunlight energy, is is light dependent. So how many ATP do we gain at the end? You don't need to know that for um what's its face photosynthesis, but you will need to know how much ATP is produced by cellular respiration, which we will go through is our very next subject. Oh yeah, and then NADPH and NADP plus. NADPH is an electron carrier, so basically what it does is it transports electrons from one process to the other. And so in NADPH form, it's carrying an electron. Well, I think yeah. So NADPH and NADP plus are electron carriers, where NADP plus is just the reduced form of NADPH. And then NADP plus goes back to the light dependent cycle, gets its electron back, turns into NADPH, and then it can come back here to the Calvin cycle, give its electron to the cycle to power it, and then it's just continue. It's pretty much the same thing that happens with ATP and ADP, except the energy provided by NADPH is in the form of electrons. Can I go through light dependent real quick? Yeah, sure. Um, so basically, photosystem 2 channels light energy to a central pair of chlorophyll atoms, releases an electron. The electron travels through the membrane of the organelle, creates a hydrogen proton gradient, meaning that there's a lot more protons inside the organelle. There's not a whole lot out here. And then photosystem 1 receives the electron, also some energy from sunlight, ejects another electron that goes down the transport chain, and binds with NADP plus to turn it back into NADPH. And then finally, ATP synthase allows the hydrogen protons to pass through it and harnesses the energy from that to turn ADP back into ATP. And these electrons that we're losing, they are replenished by breaking down water molecules into oxygen and hydrogen, which releases electrons that we can use to replace whatever it is we use. All right. Now let's do a progress check regarding photosynthesis. Your question is, which of the following best describes the primary function of a light-dependent reaction in photosynthesis? All right. Well, looks like we have three answers, all of which are A. A is correct. So the point of the light-dependent reaction is to generate ATP and NADPH, and these two molecules will power the Calvin cycle. B is not correct because B says the light-dependent reactions produce glucose. That is the job of the light-independent reaction. C is the fixed atmospheric nitrogen. That is not the point of photosynthesis at all. D is to convert glucose into pyruvate through a series of redox reactions that isn't in photosynthesis but is in cellular respiration, which we are now going to go over. So, cellular respiration, both plant cells and animal cells can do it. Animal cells are just better at it because cellular respiration happens in the mitochondria, and animal cells have more of those to make up for the fact that we can't photosynthesize. So all of our energy production comes from cellular respiration. So first is glycolysis, uh, and this is where we take a glucose molecule and we split it into two pyruvic acid molecules. This is going to give us 2 ATP, and then it's going to power the Krebs cycle in the mitochondria, and that produces carbon dioxide as a waste product. It also gives us some chemical energy that's going to power our electron transport chain. And these two processes are going to make 36 ATP for a total of 38 ATP for cellular respiration. And this 38 ATP figure is assuming the absolute best case scenario possible. So it's like no waste, uh, none of this ATP goes back into powering any of these reactions. So the absolute maximum amount of ATP we can produce from one cellular respiration cycle is 38. Now, in your body, obviously, no one is perfect. So really, every time we perform one cycle of cellular respiration, we really get a net ATP count of 30 to 32. And then we can see here that the electron transport system requires the intake of oxygen, uh, and it gives us water. All right, so let's take a closer look. So here we've got glycolysis, which is where we take one glucose molecule, we basically tear it in half, and we get two pyruvate molecules. This produces some ATP, two molecules, as you've just seen. Also gives us the electron carrier NADH. Now, step two is pyruvate oxidation. These two pyruvate molecules that we just made in glycolysis, they're going to travel through the mitochondrial membrane, which is represented here. It's a bilayer. Step two, pyruvate oxidation. Here, pyruvate is converted into a molecule called acetyl coenzyme A, or for short, acetyl CoA. And then this process produces carbon dioxide as a waste, which is why we exhale carbon dioxide. This is where it comes from. And this also produces some more of the electron carrier, NADH. Then acetyl CoA made in pyruvate oxidation goes into the citric acid cycle, where it combines with a four carbon molecule through a chain of reactions. It ultimately, sort of like photosynthesis, it's going to regenerate the four carbon starting molecule, but all these specific steps, every single molecule involved, we're not concerned about that for the scope of this exam and this course. What we're concerned about and what you need to know for the exam is acetyl CoA goes into citric acid cycle. It gives us ATP, some more waste carbon dioxide, and these two electron carriers, NADH and FADH2. And finally, all this ATP and these electron carriers, they're going to go into oxidative phosphorylation, which we're going to take an even closer look at right here. So first, we're going to all this happens inside the mitochondria. So first, that electron carrier NADH from the citric acid cycle is going to go into this embedded protein in the membrane wall. The name for this complex is just complex 1, nothing too fancy about it. But we're going to take an electron from that NADH. The NADH turns into NAD plus the proton H plus. Um, and then the electron moves into the electron transport chain. And then complex 2 does the same thing, except it uses the electron carrier FADH2, steals an electron from it, turns it into FAD, and two hydrogen protons. So these electrons all go through the transport chain through complexes 3 and 4. Um, And we're doing the same thing here as we're doing in the light-dependent reactions in photosynthesis. Our goal is to create that hydrogen proton gradient. We want a lot of hydrogen protons in the intermembrane space. So then later they can come through the ATP synthase that once again harnesses the energy of the protons coming through it to bind a phosphate group onto ATP to turn it into to bond a phosphate group to ADP to turn it into ATP, which is the main form of energy used by animal cells. And then finally, the electrons in the electron transport chain are going to go through, come out the end here. We're going to take oxygen, hydrogen, and that electron that came back out. We're going to make water out of it. So all of these are oxygen dependent reactions, meaning that they require an input of oxygen to make those vast amounts of ATP. As you can see here, oxygen is a reactant in oxidative phosphorylation. It's also in the name oxidative. But what happens when we're exercising our muscles for a really long time? We've exhausted all the dissolved oxygen being carried in by our blood. Well, we can still produce ATP, uh, but we're not going to be able to make a whole lot of it. We can do that through a process called fermentation, where glycolysis happens as usual. We take a glucose molecule, get our 2-pyruvate out of it, we produce our 2 ATP, get our 2 NADH electron carriers, but then that's it. We can't go through this whole process of oxidative phosphorylation because we have no oxygen to use. So instead, we're going to go through a kind of complex chemical process that you don't need to know everything about. It's just called NAD plus regeneration, where we're going to take some electrons from this chemical process to turn our NADH, well, sorry, the electrons from NADH, they're going to power this process, turn back into NAD plus, and they're going to go back up to retrieve more electrons from glycolysis. But at the end of this process, we're going to end up with lactate or lactic acid, which scientists thought was what made your muscles hurt after a long workout, but no, they're not so sure. But I digress. The important part here is that see that the entirety of the ATP that we make is the 2 ATP that comes from glycolysis, which when compared to the 38 ATP when we're allowed to use oxidative phosphorylation, that's like a really big difference. So essentially, fermentation is what we use when we run out of oxygen to power the super efficient oxidative phosphorylation step. Um, Fermentation allows us to keep going, but we're not going to be able to produce as much energy as we were before, and we eventually get tired. So we have to stop, rest, intake some more oxygen. So that's to the point where we can start up the oxidative phosphorylation again and start producing those 38 ATP every cycle. Yes, fermentation is going to be far less effective and efficient as oxidative phosphorylation because you can see here that um, glycolysis, every cycle of that produces 2 molecules of ATP. Whereas if we're allowed to progress through the Krebs cycle and eventually the electron transport system, that can produce 36 more ATP. So we could be making 38 total. But in anaerobic processes, we're only allowed to make two. So that's a pretty big difference. And also, primitive single cell organisms, such as bacteria, they don't have mitochondria. And since you need mitochondria to run the Krebs cycle and the electron transport system, they're not allowed to do cellular respiration. Well, they're not allowed to do aerobic cellular respiration, I should say. The entirety of their energy needs come from glycolysis. So, regarding holding your breath for extended periods of time, yeah, the reason you can't hold your breath for forever is because your body needs oxygen to, and this is why your body needs oxygen to power oxidative phosphorylation. Um, and you might be saying, well, why can't my body just do fermentation and keep making all the ATP that it needs? Well, the lactate produced lactic acid, that's a waste product. We can't do anything with that, so we've got to get rid of it somehow. But through fermentation, it builds up faster than we can get rid of it. So at some point, you're gonna have to stop fermentation to give your body a chance to get rid of all this lactate. Uh, and so the only other option we have to produce energy is to use oxygen. So you're going to have to start breathing again at some point to get more oxygen. Then what do we need fermentation for? Well, fermentation is what happens when, let's say you're outside, you're playing a nice game of soccer, uh, but the match is tied, and so you keep at it, but teams are evenly matched, so you end up dragging out the game for like an hour. Now, oxidative phosphorylation is what your body will prioritize, but it also uses oxygen pretty fast, faster than your lungs can replenish the oxygen. So once you run out of oxygen to use in oxidative phosphorylation, we're going to have to turn to fermentation because fermentation does not require oxygen. We can keep going for a while longer, but eventually we're going to build up too much lactate. We're going to have to stop, give your body a rest to recharge enough oxygen to use oxidative phosphorylation again. So essentially, fermentation takes over when we don't have enough oxygen to use aerobic cellular respiration anymore. Did that answer your question? Glycolysis happens no matter what, right? Yeah, so being the first step in cellular respiration, glycolysis is always going to happen. Now, whether the cellular respiration cycle continues into aerobic respiration depends on whether we've got oxygen present and also whether the organism in question has mitochondria. If both of those requirements are met, then we can continue into the Krebs cycle and then into the electron transfer system. If either one or both of those conditions are not met, we have to resort to fermentation, which is a lot less efficient. So if the body doesn't have enough glucose, it would just stop producing energy in total. Yeah, that's basically just starvation, starving to death, which is why you need to eat. So eating, your body breaks down your food, turns into a bunch of different things you need, like protein. But carbs are the main source of glucose in the body, and this is why you need glucose to start cellular respiration, because glycolysis is the first step that lets everything else happen. The two requirements for the aerobic portion of cellular respiration is that one, you have to have mitochondria, and that's because inside the mitochondria is where aerobic cellular respiration happens. So, like bacteria, they don't have mitochondria. They can't do aerobic respiration. And then the second one is because it's called aerobic respiration, it's in the name, oxidative phosphorylation, oxidative oxygen. You need to have sufficient stores of oxygen to perform oxidative phosphorylation. All right. Now we're going to do a progress check regarding, uh, yeah, regarding cellular respiration. So I'm pretty sure I saw a question regarding this exact substance, cyanide, when I took the exam. So the question goes, cyanide acts as a poison because it inhibits complex 4 of the electron transport chain, making it unable to transfer electrons. How would cyanide poisoning affect the electron transport chain and the proton gradient? All right. Looks like two of you think it's D, one of you think it's B. So let's see. The correct answer is D. The electron transport chain stops and the gradient becomes weaker. So to answer this question, we need to know first off what the electron transport chain is. Um, we It would help to answer the question if we knew what complex 4 was referring to, but we wouldn't necessarily 100% need it. But let's go back to the diagram here. So complex 4 refers to this one. And we can see that it helps create the proton gradient. So if um, if cyanide were to stop the functioning of complex 4, then what would happen is essentially the electrons would not be able to travel through the entire electron transport chain, and the chain would shut down. We wouldn't be able to make the proton gradient anymore, and ATP production would stop. That's how cyanide poisons you. And the reason it is not B is because we can see that the electron transport chain, it needs to go through all four of the complexes here. Uh, Electrons from NADH start in 1, go through complex Q, 3, and then 4, whereas electrons from FADH2 start in 2, go through 3 and 4. So essentially, we can't force the electron transport chain to reroute through complex 2 because that's just not what it does. And so that's why B is not the answer. C and A are not the answer because as we've just gone over, all the electrons need to finish the transport chain from 1 or 2 all the way through 3 and then finally out through 4 to complete the electron transport chain. So the correct answer here is D. So in case of cyanide poisoning, the body would start fermentation. Well, I suppose it could, but eventually you're not going to be able to make enough ATP to sustain life, which is why, what was it, cyanide eventually kills. Because we take a lot of energy to function. It's a lot more than just 2 ATP per cellular respiration cycle. All right. So the last topic in the last topic in Unit 7, crap. The last topic in Unit 3 is fitness, which I don't know why it's in Unit 3. It really seems like a Unit 7 topic lumped together with natural selection and such. But fitness describes an organism's ability to survive and reproduce. And exactly how this has to do with Unit 3 is summarized here. Different organisms will have different amounts and types of the molecules and organelles in their cells, which affects their fitness. And then if a change in the environment occurs, only the organisms with specific molecular adaptations suited to those conditions will be able to survive and reproduce and pass on their advantageous traits. So in this image on the right, you can see some moths. There are four of them in the image. The two on the left are, you could call that white, and the two on the right are black. And you see how it's really easy to notice the white moth on the black background. And it's really easy to notice the black moth on the white background. And this ties into our second point here, because we can conclude that, hey, this black moth, it probably has a lot of melanin in each of its cells, which is what gives it that dark color and lets it hide from predators on the this black dirt or rock, whatever that is. The same applies here. The increased amounts of melanin in the black moth cells, make it stand out on this pale rock. And so predators hunting for moths on the pale rock will be able to spot the black moth easily, and predators hunting for moths on the black rock can see the white moth easily. And so any moths living on a black rock, eventually over generations, they're pretty much gonna all be black moths because the black moths are gonna have a higher chance to survive, to reproduce, and pass on that trait of having more melanin in their cells. And the same applies for the pale rock. Eventually, over many generations, all the moths living on pale rocks will be pale. So, our progress check for this unit is In a population of beetles, individuals vary in shell color. Some are light brown and others are dark brown. Birds easily spot and eat light brown beetles on the dark soil. Over several generations, the frequency of dark brown beetles increases. Which of the following best explains this change? Well, it looks like we're all in agreement that the answer is B. The answer is B. Dark brown beetles have greater fitness because they are more likely to survive and reproduce. Uh, D is not correct because it says there is no selection pressure, whereas obviously there is selection pressure because the beetles are being preyed on by the birds. C is you should be able to tell that's not right off the spot. Um, a bird isn't going to know whether a beetle has more nutritional value based on its shell color, and even more so if those beetles belong to the same species. A seems right at first glance, but if you take a closer look, it says turn darker over their lifetime. Um, we should know by now that evolution doesn't happen over an organism's lifetime. It happens over many, many generations. And so that's what makes A incorrect. So that brings us to the end of the Unit 3 review for tonight. Um, if you wanted further review, here's some things that I would recommend that you do. So first is visit your AP Classroom website. You've got all sorts of progress checks, like the ones that we've been doing through the lecture. You've got AP Daily that you can watch. You've also got access to FRQs that you can practice with. Um, you can also visit the Five Hive website, which is fivehive.org. We will be posting eventually all of our practice materials, recorded lectures, and such on there. But it is very much a work in progress still. So if you do not find what you are looking for, shoot one of us a message and we'll be sure to bring it up with our team. Uh, Generative AIs like ChatGPT or Claude or whatever one you might prefer, they're advanced enough to give you pretty good preparation for standardized exams such as this. But word of caution here is that sometimes they can get things wrong. So absolutely use caution when you're using a generative AI to prepare for an exam. Always make sure that whatever it's telling you is factual. You might have to go somewhere else to verify that, do a quick Google search or what something like that. And then finally, old reliable Khan Academy. Highly recommend it. That's where I got some of the diagrams in this lecture from, actually. So, um, yeah, and did I mention all of these? All of these resources are free, except if you use a GPT subscription. Obviously, that's not free. So, anyways, just another reminder to please fill out the survey that I talked about at the beginning of this lecture. It helps us tailor our lectures and content to better suit the needs of AP students. So, that's you right now. Could be your siblings later, or your friends, or anyone. Uh, you don't have to know them, but any feedback helps. At this point, I'm going to stop the recording. Thank you all for coming. If at this point you do still have any questions regarding the Unit 3 content, now is the time to ask.